In today's lecture, we're going to cover the transcription translation. So what's transcription? What's translation? We're going to look more broader. Okay. So the nucleus and the DNA. Let's first of all talk about this, about the nucleus and what's DNA. The DNA is what? It's a genetic material, right? It stores all the instructions a cell needs to function. It contains all the instructions so the cell should do. It's like a recipe for the cell to do its job. Okay, everything is written in the DNA. Okay, in eukaryotic cells, for example, in the human cells, and DNA is packaged inside of the membrane-bound nucleus. It's packaged inside of the nucleus in eukaryotes. In prokaryotic cells, in bacteria, for example, they have also the DNA, but they don't have the nucleus in the prokaryotes, right? Their DNA is circular in shape. In bacteria, their DNA is circular in shape, and they are located in the region of the cytoplasm, which is called what? Which is called nucleoid. Okay, nucleoid is a region which is located in the cytoplasm where the DNA is located. Okay, in the prokaryotes, and the DNA in the prokaryotes still has the double helix structure. They all have thymine, adenine, cytosine, and guanine, and in the bacterial cell as well, they have the complementarity rule. Okay, so in the eukaryotic cells, we have the nucleus. In prokaryotes, we don't have the nucleus. Okay, DNA in in both structures, in both eukaryotic and prokaryotes, they have the DNA structure. DNA in eukaryotes located inside of the DNA in eukaryotes located inside of the nucleus, but DNA in prokaryotes located inside of the nucleoid, which is in the cytoplasm. Okay, and the chromosome shape. In the eukaryotes, you see the chromosomal shape is linear in shape, but in bacteria, they are circular in shape. Okay, the DNA is circular in shape. It's like this. So, and also, okay, they are circular in shape, the bacterial DNA. Okay, so DNA blocks, you know the blocks of the DNA, adenine, cytosine, thymine, guanine, you know all of them. Okay, so the cell is like a library. The nucleus is a special archive room. This archive, nucleus is archive in eukaryotic cells. Okay, in bacteria, the books, which we book, it's a DNA actually. These books are just on the shelves on the open space, still readable, but not locked away. But in eukaryotic cells, the books are locked away in the library, which we call the nucleus. Okay, good. Any questions? No? You see, in the nucleus, we have the chromosomes, and the chromosomes contain the DNA, which is condensed. Okay, which is condensed. So DNA replication. What's DNA replication? What do you understand by DNA replication?
Increasing in number. For example, from one DNA, it makes two.
Yes, from one DNA, they are making two DNAs, and this is called what? DNA replication. So DNA means making a copy of the DNA, so the cell can divide and give each new cell the same genetic information. So from one cell, we are going to make two cells, right? So each cell must contain the same gene, the same DNA, like a previous one, like a previous one. In order to make and get the previous genes, the DNA in the parent cell, in the previous cell, must do DNA replication, must do copying of its DNA. DNA replication means copying of DNA, making a copy of the DNA, okay? Its DNA is a zipper, okay, made of letters. They are made of adenine, thymine, guanine, and cytosine, okay? Replication is like unzipping it, okay? So DNA is like zipper with a teeth which has a letters adenine, thymine, guanine, cytosine, but replication is unzipping it and building a matching new zipper on both sides. Do you understand me? Right? First of all, there are steps there are steps. First step is called for the replication unwinding. What's unwinding? The DNA double helix unzips into two single strands. This is called what? Unwinding. Imagine this DNA molecule, right? It's going to become like this, unwinding. This process is called what? Unwinding. You're separating them into two single strands, and this process is called what? Unwinding process. Unzipping a jacket, it's like unzipping the jacket. Then you're going to match the bases. We have a special enzyme which is called DNA polymerase. Can you see this DNA polymerase? This is the DNA polymerase. Is it visible?
Yes.
Yes,
Yes.
this DNA polymerase is going to start here, and it's going to move along the DNA. It's going to move along the DNA, moving along the DNA. So when it moves along the DNA, what it's going to do, it's going to make the new strand of the DNA. It's going to make the new strand of the DNA. Now, this is my one DNA, this is second DNA. You see, from one this previous DNA, I'm getting two new DNAs.
Mhm.
Okay, from one, I'm unwinding, I'm separating strands, and from each strand, I'm making the new strand with the help of DNA polymerase. DNA polymerase means making DNA from DNA. Okay, making the new strand of the DNA from the DNA. So I'm making the new strand according to the complementarity. If I have the adenine here, if I have the adenine here, the next strand will be, sorry, thymine. If I have guanine, next strand will be cytosine. If we have guanine, the next strand is according to the complementarity, they're going to build up, they're going to make the new strand of DNA. Okay? Do you get my point? Do you get what I
Yes.
mean? Okay, good. From one, now I have to matching bases. It's called matching bases. Adenine matching with thymine, guanine matching with guanine. Okay, so then what's going to happen? The bond formation. What's bond formation? Hydrogen bonds should form between the base pairs, right? And the sugar phosphate backbone should connect the sides. This backbone should connect two sides, the phosphate backbones, right? So hydrogen bonds are going to now form between adenine and thymine. Two hydrogen bonds, cytosine, guanine, three hydrogen bonds are going to form. Okay. Then twisting back, what they should do, they must twist now. Again, they must twist. They must get this shape again. Okay, they're going to twist. We have the special enzymes which are called topoisomerases. Those topoisomerases are going to what? Twist it back. It's going to twist the double helices back. Okay, now you have two identical zippers, one for each cell. Even though when you divide the cell, when you will have two cells, you're going to have two exact information because all the information came from this DNA. Okay, any questions so far?
Mm.
So what's going on here in the DNA replication here? You see the parent DNA, right? This is called what? Parent DNA. This is the original double-stranded DNA. One dark blue, okay? This is the dark blue in color, okay? This is called what? Old strand, or we call it parent strand. And we have the daughter DNAs. So daughter DNA means this is a new DNA molecule formed after the replication, okay? You see, this is the new strand, okay? This is we call new daughter DNAs. They are new DNAs which is synthesized from the previous ones, okay? We call also it new strand. This is strand built by the enzymes, which we call pink, okay? And we have the old strand, which is template strand from the parent DNA, okay? So direction of the replication is from the 5 prime to the 3 prime. From the 5 prime, if you see this 5 prime, it's starting, the DNA polymerase is here. It's going to synthesize the new pink strand from 5 prime to 3 prime. Okay, from 5 prime to 3 prime, they're going to synthesize it. Okay, after replication, each new DNA has one old strand. Each new DNA will have the old strand. This is the dark bluish color, and the new strand, which is pinkish strand. The new strand is built by which enzyme?
Then the
Of course, DNA polymerase. It's called DNA polymerase. Good. So I hope you understood it, right? This is very easy process, DNA replication. So what's primer? Here I forgot to tell about you about the primer. What's primer? Primer is where the DNA polymerase sits in the first, okay? The primer, to the primer, first of all, DNA polymerase is going to start. This is the origin of replication. This is the start of the replication. First of all, DNA polymerase is going to sit to the primer, and then it's going to move primer. This is a set of nucleotides where the DNA polymerase is going to attach to start the DNA replication. Okay? This is the primer. This is the place where DNA polymerase first sit. Any questions? No? Okay, how many chromosomes in the human cell? How many chromosomes do you have?
46.
Yes, 46 chromosomes. So in a normal human body, all of them have 46 chromosomes, right? So 46, this is a diploid number. So chromosomes, they are bundles of the DNA. So chromosomes made of the DNA, okay? They carry all the instructions with genes that your body needs to function, right? So you have types of the cells in your body. What types of the cells in your body do you have? You have the somatic cells. Somatic cells. Okay, what's the meaning of somatic cells? Somatic cells, and you have the gametes. And you have the gametes. So somatic cells means non-sex cells. Non-sex cells. What's the meaning of non-sex cells? Non-sex cells, they have 46 chromosomes in the humans, okay? This number is called diploid in number, and these chromosomes are organized in pairs. Do you see them? The paired organized chromosomes, they are somatic cell chromosomes. All of them are non-sex chromosomes. They are diploid in number, 46 chromosomes. At the end, you only have sex chromosomes. Okay, it's in. So you have the somatic cell and gametes. Meaning of the gametes, it can be either sperm or it can be the ovum. Okay, ovum and the sperm, they have 23 chromosomes. In the somatic cells, they have 46 chromosomes, but in gametes or in the egg or in the cell, they have 23 chromosomes, which is a haploid in number. We call them haploid in number, so we indicate them as an N, and these ones are 2N. They are diploid in number, I mean the somatic cells. All cells in your body have the same set of the chromosomes. So your skin, your kidney, your brain, they have the identical chromosomes. If I take your brain cells and if I take your skin cells, they're going to look identical. Okay, their chromosomes are going to have, their cells going to have the same number of the chromosomes, same shape of the chromosomes. Okay, they're going to be identical. You have your identical chromosomes. So what about identical twins? Do you know what's twin? Близнецы, they have the same set of the chromosomes. So sorry, they came from the same egg. They came same egg and the same sperm. So their chromosomes are 100% identical. This is like natural cloning. Okay, identical twins, they are natural cloning. They came from one egg and one sperm. That's why their chromosomes, even though they are two organisms, their chromosomes look like to each other, similar to each other. But my chromosomes and your chromosomes, they're going to differ. But your own organism chromosomes, your brain and your skins, they are from one organism, they are exactly the same. Okay? So other organisms like nematodes, here nematodes, they have fewer chromosomes. Their diploid number is 4. In this picture, you can see their diploid number is 4. Their haploid number is 2 in nematodes. Okay. So how to find the haploid number? You just divide the diploid number into 2, and you will get the haploid number of the chromosomes. Okay. So identical twin, there is one fact: one fertilized egg is going to split in two. Imagine you have one fertilized egg, one sperm entered, okay, it fertilized, you have the one fertilized egg, оплодотворенные яйцеклетки. What happens in the identical twins? They're just going to divide into two. This fertilized egg just divides into two, okay? They both carry the exact same DNA because they came from the same original chromosome. They came from the egg and the sperm. This is why identical twins look like alike to each other. Okay, look alike to each other and have the same genetic trait. They have the same genetic chromosome. Okay, good. Any questions? Imagine this is like what's analogy to it. Imagine each cell in your body, each cell in your body has a complete instruction manual with 46 pages. Okay, each cell in your body. But in your gametes, in your egg cell, for example, or in the sperm cell, they only carry half of that page, only 23 pages. So when they combine, so 23 pages of the father, 23 pages of the mother, when they're going to combine, they're going to make 46-page organism. Okay, so full 46-page book is revealed in the embryo. Okay, understandable? Any questions regarding that? Sağ Now, what are the homologous chromosomes? Homologous chromosomes, they are pairs of the chromosomes, particular chromosomes with their pairs. Okay, this is called homologous chromosomes. One came from your mom, one came from your dad. They are same in their lengths, they are same, they have the same genes in the same location of the chromosome, but might have different versions of the gene. They are called the alleles. Okay. So homologous chromosome here, this is one chromosome, this is another chromosome, this is pair of the chromosome. How many chromosomes totally? Here we have
2.
Не понял. How many chromosomes we have here?
46.
44 and 45. 46.
36?
46.
46, yes, correct, 46. How many pairs?
22.
22. No,
44.
How many pairs of the chromosomes are there?
44. The last one isn't fair, right?
22.
22 pairs, almost 20, it should be 23, but this is Y chromosome. What's the meaning of Y chromosome?
It's a of chromosome of male.
Yes, this is male organism chromosome. Okay, this is X. This came from the mom. Sorry, this came from mom. This came from dad. Okay, and we have 22 autosomes. Autosomes means non-sex chromosomes. 22 pairs. They are autosomes. Pairs. 22 pairs. They are autosomes. They are non-sex chromosomes. But one pair is sex chromosome. One came from mom. One came from dad. Y came from dad. Okay. So each pair here, each pair came from dad, mom, okay? They are same in their length, same in their gene location, but the gene version is different. For example, here in the same place, we have two places here. In this place, you have a gene which is responsible for the eye color, which is brown, but here it will be eye color, which is responsible for the blue eye, okay? So they contain the same genes, but their versions different. Okay, they called homologous pairs. Same in their length, shape, same in their gene and their location, but different in the gene what versions. Okay, so homologous pairs, they are called homologous pairs, exactly the same pair of the chromosome. Okay, so what's diploid number? Cells with two sets of the chromosome is called, there are two sets. One set of chromosome is 23 chromosomes. They are in gametes. But two set of the chromosome is 46. They are in somatic cells, all your cells except gametes. Okay, so haploid means one set of the chromosome, egg cell or sperm cell. Fertilization means sperm cell with 23 chromosomes and the egg cell with 23 chromosomes. They're going to combine to form 46 chromosomes in the new zygote. Okay, so uh, each pair of set of instructions, mom gives you one copy, dad is going to give you one copy, mom is going to give you 23 pages, dad is going to give you 23 pages of instructions. So you will have what? At the end, 46 pages, okay? So gene X, gene Y, you know the differences, okay? So any questions regarding this?
Mm-mm.
Good, let's do this task. Who starts? Who will start?